I am currently studying the regulation of the MUB gene by vitamin A. In this study, cloning vitamin A receptor is necessary for studying it. To sequence a piece of DNA, I need to use cloning to produce many pieces of the gene that I'm interested in studying. I insert the gene into E. coli bacteria and then put them into a plate. That will allow the bacteria to grow rapidly. So overnight, many copies of the DNA will be produced. After extracting the DNA from the bacteria, I need to make sure the clone made is correct by DNA sequencing. To prepare the sample to go to the core facility, first, I need to add to a tube DNA, primers, and water, and then mix them gently by pipetting up and down several times. Now the sample can be sent for sequencing. Only if the DNA sequence is 100% correct as predicted, the protein produced based on this DNA sequence will be expressed and function as expected. Here is the sequencing result for my DNA from the automated DNA sequencer at USC DNA Corp facility.